It is now my absolute honor to invite Dr. Barry Karger to the podium and we can celebrate him on his way up, and we can then listen to him. Dr. Karger.
Oh, sit down. Well, I want to welcome everybody here. Most of the people here I actually know, and I'll have to say hello during the day. But I also want to welcome all the people online. First of all, Elliot and Laurie, and sorry you can't be here, but I hope soon after this we arrange so we can have a Zoom call together. And I want to also welcome all the people online from all over the world, from Latin America, South America, Europe, Israel, Mumbai, India, and Hong Kong, for example, all former students. My first slide was actually much later in my talk, but I put it first because it's the most important one. I want to recognize all the Barnett alumni. You know, when you think in terms of what you're most proud of, and now I speak for all the faculty fellows, and your most important contribution, it's all the students you put out. And over the years, the Barnett Institute has put out over 500 PhDs, postdocs, and staff, all had at least a PhD in the staff. And many of them, most of them have gone on to distinguished careers, and it's quite a group here today. We've had academic leaders, CEOs. I wish I had time to talk about the vice presidents are here, directors in pharma, biotech, and other areas, clinical diagnostics, instrumentation companies, venture capitalists, investment people, and I think it's fair to say that through these students we've had a major impact in biotechnology. And we're most proud of all of our alumni. Now I'm going to take a few names as I give a talk, and I apologize to all the others, but they didn't give me enough time. So you can blame our future leadership. Okay. But before I begin, Henry James was a famous author of the 19th century, American and British, and he said, "A teacher affects eternity. He," and now I add she, "can never tell where her influence stops." And what was really remarkable, on May 1st, Nature had a memorial article about Alan Bard, who passed away in February, and he said about the same thing when he got the Priestley Medal. He says, well, we're all going to be forgotten, but we're going to be remembered by our students. And one other saying I want to put in, the pursuit of knowledge is a lifelong journey, one that requires curiosity, humility, and an open mind. And I want to emphasize open mind. And this is a key point. You can't be afraid of doing new things. And science, and this is what Hazel said, science changes so rapidly. Each one of you, and I've had to do that, you're going to have to reinvent yourself multiple times. And you can't be afraid of new things. Right now, you'd have to learn algorithms and machine learning and so forth, and who knows in 5 or 10 years. But that's very important. Now, one alumnus I would like to recognize initially is Doug DeVivo, who actually is not an alumnus of the Institute because he graduated, got his doctorate with me in 1970. Nevertheless, he worked in industry and then went into investing and venture capital, and he's a partner of ALS Investors. And over the years, Doug supported our graduate students. We gave graduate students cash awards because when he was a graduate student, he didn't have enough money. And you know what's going on now with unionization and so forth, but he was there at an early stage. And I want to also tell everybody he made a generous gift to help support this program. And for all of us, I want to say thanks, Doug. It was wonderful talking with you. Now, in 1973, I was already here 10 years. Northeastern is a little different than it is today. More than a little, major difference. It was a commuter school. The University of Massachusetts in Boston had started 10 years earlier, but it still was growing and so forth. And this was really the commuter school. It was easy to get in. I put 80%, it might have been more like 90%, but to get in. Now, boy. I actually found, after some archaeology, $1,890 was the tuition. All right? And that's about $30,000 today. So nevertheless, this doesn't have fees, and of course, most people lived at home. The PhD programs began in the early 60s, and it's fair to say Northeastern was a minor research university. My wife and I come from Boston. Now you know why we came to Northeastern after Cornell. But Northeastern had an important point, work-study. And this I'm going to emphasize again in a minute. Now, I and others, and I want to recognize Bob Lowndes, who worked with me. We worked with President Ken Ryder and Executive VP Jack Curry, who then became president, to strengthen the research environment in Northeastern. We helped put a person in the provost's office for research, a research council, and many other things over the years. What happened here? All right. Can you see it? Okay, good enough. In any event, 1973, there was no internet. You had to go to the library and hope that it wasn't checked out, the book. There was no laptop, no cell phones. Now this is important. Ties to industry were either limited or non-existent. And if you did applied work, well, you were at the lower end of the scale. And most of the faculty were solo. Collaboration was really unusual or very limited. I put in a couple of things, on the science. Maybe this was more in the 60s, but I remember when I used to try to quantitate chromatograms, we'd have to cut out the peaks and weigh them, and we worried how much water was in the paper. Or we used a planimeter. And then at that time, GCMS was being done, and in order to do polar compounds, there was a lot of exciting work, very sophisticated work in derivatization. But LC was not yet developed. And this was the Dark Ages. But this is a very important point. We had time to think. We didn't have so many emails. And so I look back on that time, oh, that was so wonderful. All right, now, there's a lot of discussion about analytical chemistry, and I had an interview for a start of a meeting about the prestige of analytical now, but no time to talk about that here. But the way we see it is analytical is at the center, and the spokes on the outside of the applications. So you have clinical, therapeutics, forensics, et cetera, et cetera. And the analytical is key. If we take pharmaceutical and biotechnology, I mean, if you can't analyze your compound and know what you have nothing. You'll never get it approved. So this is a fundamental thing the analytical. All right, so now, how did the Institute get started? Well, I had a sabbatical in the fall of the 72-73 academic year. In the Department of Chemistry, at that time it was called the Department of Chemistry, the Dreyfus Foundation, they felt, had given them a hint that, gee, if you come up with an idea, we might be willing to fund it. So they had meetings, and they said, gee, yeah, we ought to do it. And then they said, who's going to do it? Well, the person who wasn't in the room, obviously. So that was me. But I had, was thinking that way anyway. And so we went ahead, and Northeastern, because of the work study, had a College of Criminal Justice. It had at that time, a College of Pharmacy. And also the engineering school. It was the #2 engineering school in the Boston area after MIT. And so I talked to the deans of these colleges and Dean Rosenblatt of the criminal justice, and we applied to the Department of Justice. They were putting together a 7-school consortium to try to upgrade criminal justice personnel and so forth. And by Jove, we got the grant. And that started the Institute of Chemical Analysis Applications Forensic Science. It's a mouthful, I agree. So I was the director, and Bill Geeson, who unfortunately passed away in 2010, was associate director. And we worked together, and he was really very helpful in the growth of the Institute. Now, what's very important is, from day one, the culture was that we would do both fundamental and applied, and the sexy word now is translational, but it's applied, technology to the real world. We didn't just want to develop fundamental and then say, okay, now what are we going to do with it? We said we wanted to understand the problems, and then if necessary to have fundamental, fine, and go ahead. And many of you know I always say, if you're going to be an academic scientist, you want 1 foot in NIH and 1 foot in industry, medical school, or what have you. The second thing is, we understood that we didn't know everything. It was very important to do collaborations. Number 3, entrepreneurial. 4, we felt it was important, if we're going to have impact, to do training. And number 5, as I've already said, we understood we're going to have to keep reinventing ourselves. And this is an important point, and I realized it even more as I was putting this talk together. We had a big advantage because the culture at Northeastern University was in that direction, even back at that time when many people said, if you do applied work, well, you're lower level scientists and so forth, because of the work study. And of course, today, everybody's doing this. So we were just way ahead of the game. All right, so quickly, in forensic science, we worked until 1976 when Jimmy Carter came in from Gerald Ford, and the Department of Justice stopped the program at that, but we continued. You can see, one thing is there was a famous trial, Howard Hughes' will. We actually analyzed the ink for that at that time to try to check, and one of our people testified and explained what chromatography is at the trial. But this continues forensic till today, when the dean, Norm Rosenblatt, passed away in 1990. We established a lectureship once a year, and then one of the speakers was Dick Saperstein, very well known in forensic science. He's passed away, and he endowed the lectureship, which continues to today. And all the top people named in forensic science, even someone who analyzed the DNA for the bones of the Tsar's family in Russia, et cetera, have spoken in that. All right, now in 1973, so it is when we began the Institute, the book An Introduction to Separation Science came up. It was co-authored by myself, Lloyd Snyder, and Csaba Horváth. They're both giants in the field of chromatography. So separation fundamentals, interesting for the people in the audience. Mass spectrometry was only 2 pages. But this was a graduate text used for over 25 years. Now, Csaba Horváth was a professor in chemical engineering at Yale. And by the way, he got into the National Academy of Engineering. And Csaba and I, since the location, used to have twice a year symposia, once down at New Haven and once up here. And then, you know, beer and pizza after that. So our 2 groups knew each other pretty well. So I put 2 names down who are here today. They came in from California. Wassim was a postdoc with me, and Stacy got her PhD with Csaba from at Yale. So Stacy was at Genentech, and then Wassim went to Genentech, and they actually got married. And Wassim is a vice president of Genentech and is on the management board and the representative of biotechnology for the International Council on Harmonization. This is a group that, for regulatory, they try to make things similar from one country to another. And Stacy was vice president at Genentech. She's now executive vice president at Gilead. And I just, since it's a story, I thought it would be good that if it wasn't for Csaba or me, who knows what would have happened. All right, so in the 70s and then in the 80s and 90s, one of the things that certainly the Institute contributed to, not only by my group but others, is the development of LC. Of course, there are other groups that did it too. We helped in developing reverse phase columns, high-impact chromatography, how to pack columns, hydrophobic interaction chromatography, where you now can collect things, proteins in the active state. And of course, this is widely used in purification for bioprocessing, and more recently, sensitivity that Hazel mentioned, and certainly protein separation. So one of the contributions certainly of the Institute is liquid chromatography, and you know, after all, LC-MS, what would you do without it today? All right, one other point though is this is a chromatogram that we published in 1979. It was the first separation of chiral species by liquid chromatography. And the reason I show it isn't only that was the first one, but Wolfgang Lindner was a postdoc with me at that time. He then, after a while, became the chair of the Institute of Analytical Chemistry at the University of Vienna. In 1970s, to separate chiral species was an art, not a science. But he and two or three other people made it a science. And columns that he developed are still being used today. And without that, some proteins would not be available today. What happened here? All right. Okay, so let me, basically what this slide is, I don't know what happened, but something's going on. But the other thing is, in the 1970s, we were doing training. At that time, we were training FDA people from local labs and from Washington on liquid chromatography, how to do it. We actually had labs and so forth. And then we continued with other training, and when biotechnology began, we helped the companies develop peptide mapping and other things, characterization of the products. And then this continued, and even we trained the FDA examiners going to Washington and so forth. And then from this development, we now have a battle where there's major training going on. Jared O'Clair later this afternoon will be talking about his program, which developed from this. But I really wanted to also put in Olga Vitek, who for 10 years now has been doing training in computation and statistics for mass spectrometry. And in fact, today is the last day of a 2-week training program. And so that certainly is a contribution of the Institute, training of different groups of people. All right, in 1974, two major figures in the Institute, Roger Giese, who came as a faculty member in the College of Pharmacy at that time, and Paul Voris, who I hired to come to be a staff scientist in the Institute. And of course, he then went on to become a full professor in chemistry. Astonishingly, not amazing, astonishingly, Roger Giese is still on the faculty, teaches and so forth. But more like all of us, Paul Voris is an emeritus professor. Roger took his talents in organic chemistry into analysis, and of course, everybody knows Paul for all the work he's did in mass spectrometry. Now, I just wanted to pick one thing that both of them worked on, which has still a potential that it could be a major contribution. You know, with our environment, with different possibilities with drugs, UV radiation, et cetera, we can have modifications of DNA. And these can be DNA adducts. And this can cause cancer, either through the adducts themselves or through then mutations that can occur. And if one can analyze this, one can then be able to diagnose potential environmental or what have you cases where you could get cancer. This would be before you actually get the cancer. And this continues to today, and Roger has some derivatizing agents that are very interesting, and he's hoping to be able to spin this out into a company. But so there is a lot of potential in this area. All right, other faculty that I list here till the time I became emeritus. Philip Kane was going to be here today. Unfortunately, he has COVID, and he's home, and we wish him well. He was associate director for a time of operations with the Institute. Ira Krull, is Ira here? Oh, he was going to be here. Anyway, Rohan Matra got his PhD with Ira, and of course, Olga is here. She actually was a faculty fellow while I was still director. And we'll have more to say in a little bit. All right, now, you've heard a lot about Lou and Madlyn. Lou got his undergraduate degree here in 1944. And Gene Rapucci, is Gene here? Okay. Gene was vice president of development at that time. And we made a proposal to Lou to support, in 1981, to support young scientists. And Lou always was interested in supporting people. And so we got a grant in 1981. And from that and the relationship, in 1983, there was a naming gift. And so this was the first endowment of a center at Northeastern. And I understand from David Luzzi last night that we were the only one for a very large number of years. And what Lou said at the time was, he's not giving a gift. He's making an investment. And I'm going to say this again. And every time you get support, and that's very important, to not look at it as a gift. We're not a charity. It's an investment. And I have to say that this was one of the most wonderful things for Trudy and myself, the relationships that they developed. We really feel it's like we were like family. And we attended events back and forth. Lou and Madlyn came to the wedding of one of my daughters, and we went to their 50th anniversary and so forth. And they continue to support us, and you'll hear a little bit later about regulatory science, and at that point, a gift was made, and it really, again, was an investment that's paid off. And so I think it's wonderful. It gives me many memories, and it was nice that we talked last night. Now, you talked about how he felt this was the best, and you said this last night, but I strategically didn't say, because I wanted to surprise you. I have a big surprise. You know, I had to do archaeology to find photographs and so forth, and I found a very special one. Every 5 years, we had a celebration. And this was our 35th anniversary. And our new president, Aoun, invited Lou and Madlyn to the stage to, you know, honor them and so forth. And Madlyn, as she was so used to, said, "President Aoun, please give me the microphone." Now, if you know President Aoun, he was stunned. Nevertheless, she took it. And what she did, and this is a picture of her actually doing it, Rhoda. Okay. She said, "I want everybody to know that Lou is given, you know, been very generous to many places." He said the one place that he is most proud of and gets the most joy and so forth was the Barnett Institute. And this is the actual photograph of that. I didn't want to tell you last night, okay? All right, so the 1980s was a transition time because in 1980, we had the Bayh-Dole Act. That was a key thing. Okay, I'll go on. You know, I feel like I'm at a company interview, you know, and everybody's challenging me. Okay, all right. And, you know, at that point, universities were allowed, if they got government money, to be able to go ahead and license and then start companies. And so Bill Geeson started a company, Cambridge Analytical Associates. He was the founder, I was an advisor, which was successful, environmental and forensic. And actually now there's a faculty fellow, Zia Tian, who is working in this direction. So we've had a number of spin-outs over the years. Now, not all of them have been successful, but a number of them have. Bioseparations was a training program I did with Fred Renier, and we went to different companies at that time in the 80s because, you know, biotech, you know, they had to be trained, the people. And some of you know bioanalytics that we started in 2012. I also put in two from 2020. So there's a continual point here where we've had spin-outs. All right. I want to recognize other, really? Already? Okay. Okay. Jim Waters was, you know, very well known by everybody, and he endowed, and I was honored to have his chair for 31 years, and then John Engen has it today. In the 80s, we had biotechnology and so forth, but I really want to say we had the Human Genome Project, and perhaps of the 50 years, the thing that we most known for is our contributions in the Human Genome Project. We were able to show, we were the ones who showed that if you blew out the polymer and reloaded it, you could do a next sequence, and that was a key point for automation. And then we got to 1,300 bases. 40 to 50% of the human genome was sequenced with our technology, and it would have been 100% if the instruments could have gone to 60 or 70 degrees. $3 billion then, $500 today. And at that time, we thought, okay, RNA is just, a messenger. But damn it, it is key, exciting. All right, Peter Barrett was someone, who was executive VP at Celera. He was also with Craig Venter. The two of them were really the key to the success of Celera. And then he came to Boston. He's with Atlas Ventures. And the reason I put him also up because he was really advised us and has helped a tremendous amount. And he's also an alumnus. 1979, he got his doctorate in analytical chemistry here. Okay, then Bradstreet Chair, Raymond and Claire Bradstreet. Third one on the left is Raymond. He and his wife passed away in 92. 96, Roger and Paul were the chairs, and this was a way to bring in Bill Hancock. Most of you know Bill, and Olga, and it's wonderful that you have that. So Bill Hancock came here in the early 2000s. He's well known by everybody, and he started CAS, which now is well known, and he's even started WCBP, Well-Characterized Biopharmaceutical. There's an award, Hancock Award, for CMC Regulatory Science. Of course, he was the editor of Journal Proteome Research. And by the way, in the Hancock Award, two of the awardees have been from the Barnett Institute. One is Wassim, and the second one is Rob Garnick, who, if I have time, I hope to say something else about. So regulatory science, in other words, this is all the science to get the damn drug approved by the regulatory agency, FDA, EMA, and so forth. So at that time in 2007-8, biosimilars were coming along, but no one had the term yet. So we had used biogenerics. We got criticized by the biotech community for that, but nevertheless, so we had a meeting on this, talking about biosimilars, and that's got us going. And of course, today, many of you here are focused on CMC, chemistry, manufacturing, control, and analytical plays a critical role in this. And let me now mention Rob Garnick. He was executive VP for Genentech and left the company when Roche bought it full out in 1999. He's a major figure in this area, and through his work at Genentech, and he's a good friend, and I think a lot of people know him, and he's had a major impact in this whole field. And Jared will talk about this later. And as we go to new modalities, like now all the RNA therapeutics and so forth, there are major challenges. And if you don't have the analytical methods, you're dead. You're not going to get anything, so this is critical. All right, very good. This is, you have only about a 10% chance, maybe a little bit more now, of success from when you start in the clinic. So there's a real need to have good science in order to be able to have success with a company. John Hatsopoulos, he is the brother of George Hatsopoulos, the founder of Thermo, and at that time Thermo Electron. And with our 25th anniversary in 1998, he endowed a scholar program that, and our Jonathan Bones, who will speak this morning, When he was with us for 2 years, he was one of the Hatsopoulos scholars. And I want to say Dieter Hearn is another person who helped us tremendously. He was the head of the Hewlett-Packard Analytical Division when he retired in 1995, and that became Agilent in 1999. He was a loyal member of the Barnett Advisory Committee, and he supported lectures from 1999 for over a decade. And what we did here, and this was part of our philosophy, we had 2 speakers, one from industry and one from academia. And here, this is the second year, Lee Anderson and John Yates on proteomics. And in the 5th or 6th year, in metabolomics, we had Gary Suzak, who's well known, and then the CEO of Metabolome. So every year we took a topic, industry and academic. Let me also say, here I go. I'm almost done. I have to also say John Engen had a major contribution with hydrogen-deuterium exchange to look at the higher order of proteins and binding things, and this is well known, and he worked with Waters. More research highlights. I have 2 slides left. This is one of them. You'll hear from Olga about her work, and that's certainly part of the Barnett Institute. Nikolai Slavov, which I think is here now. Nikolai? Oh, way over there. Okay, hi, good morning. Single-cell proteomics, and that's a major contribution. You'll hear from Jared. Top-down, where you look at the intact proteins, Jeff Agar and Alexander. Alexander's an alumnus. He was a postdoc with me in the early 2000s. And then you see the others here. It's my final comments. You can applaud now. The first thing is Albert Bourla, who's of course the CEO and chairman of the board of Pfizer, has said that the next 10 and 20 years will be a golden age of life science. And I think that's going to happen. Certainly we're seeing already artificial intelligence playing a role in drug discovery. We're going to have a nice panel discussion this afternoon. But in addition, it's also for the clinical and manufacturing. It is having already a major impact in these areas. We're going to have now, the next 20 years, an era of biology and therapeutics for RNA. We already have this. And we don't have all the tools right now. The modifications of RNA, most of you know there was a National Academy Sciences report dealing with this. So there are big opportunities for the Barnett Institute. And I want to say, and this is very important, I was very happy when Olga was selected. And first of all, she's an excellent scientist. Secondly, she's in the College of Computer Science. And this is very important. And thirdly, and she heard me say this last night, she's very nice. And finally, the last thing, and I have this always at the end of my talk, I wish I were younger. It's all right.